I started doing this morning. It's going to be a little long, so I'm going to split it in two. Today will be the review of the year up to energy in the cell, and then tomorrow I'll post the other second half of this. Not sure what the final assessment is going to look like. I'll let you know that tomorrow. I'll post this today and for both classes. So we're going to review the year, what we've learned this year. And I hope you really believe and I hope you really feel that you have learned a lot this year, because we have covered, even before the virus hit, we had done a lot of hard work, important work to bring you up to speed in biology. So we started right out with Chapter 1. Chapter 1, we learned that biology is the science of life. I know I made a big deal out of the difference between science and studying, but it's important because all the information you're getting about Corona-19 virus is based on what we know of biology. It's been studied using the same types of material, the same ideas, the same concepts that we have been covering all year long. People that are doing that are known as biologists. Some of them are virologists because they specialize in viruses. Others are physicians because they specialize in treating humans, but they are all under the umbrella of biologists. They people who are studying the structure and function of living things, their history, the interaction with the environment, and any aspect of life. We also learned that organisms have some features. We had a hard time describing what life was, so we had a checklist, and on that checklist were one or more cells, displays organization, grows, develops, reproduces, responds to stimuli, use energy, maintains homeostasis, and have adaptations that evolve over time. So here at the end of the year, we've been looking more closely at how those organizations, the growth and development, the ability to reproduce, respond to stimuli, use energy, maintain homeostasis, and have adaptations that help them in particular ecosystems, biomes, or niches, allows that to occur. So it really does all come back to what we learned right in Chapter 1. We talked about what a science was, comparing things that were science with pseudoscience, and right now we are being faced with, in many cases, questions that come down to science versus pseudoscience, things that will help with COVID and things that may or may not help with COVID until proven. So, and the other thing we talked about in Chapter 1 was the use of the metric system, which can be difficult for people that are used to using another system, the American system, and SI units. But when we work in science, we work in the metric system because most of the globe does use the metric system. And it makes it easier for comparison of results. So that scientific method included asking a question, forming a hypothesis, collecting the data, analyzing the data, and reporting the results. And when done to its finest, the best way to do that, you would get what is known as a controlled experiment. So in a controlled experiment, you ask a question, form your hypothesis, what you think the answer to that question is, divide your subjects into two groups, and those two groups should be identical except for one difference, and then you collect and report the results that you get, which will either support or not support your hypothesis. Then we jump right up to Chapter 6. We come back to Chapter 2 and 3 here later on. I'll get into that tomorrow. But Chapter 6 told us what life was built from, and life is made of matter. Matter is the study of chemistry. So in Chapter 6, the important part of Chapter 6 was actually the last part of Chapter 6. The first couple of parts were just basic chemistry, and I'm sure you had covered that before. But what we really should take away from Chapter 6 is that life is built of carbon-based molecules. Carbon is very important for living organisms. Water is important, but it's the carbon molecules in a water environment that provide for life. And those carbon molecules in a watery environment are made up of 4 major families of biological molecules, and we looked at the function of each of those biological molecules. And we compared them to trains or chains, because these 4 molecules are polymers. They're long chains of individual units. The first one we looked at was the carbohydrates. Carbohydrates were made up of repeating monosaccharides, carbohydrates like glucose, starch, fructose. The second one we looked at were the lipids. And lipids were made up of individual carbon molecules, long chains of carbon molecules with very little oxygen, so they don't like to mix with water. The third one we looked at was probably the most important one. Those are the proteins. And proteins are made up of repeating units of amino acids. The information about those proteins is stored in the 4th type of molecule, and that was the nucleic acids, the RNA and DNA. They're repeating units of nucleotides. So we use those polymers, those carbon-based polymers. If we want to use energy, that will be carbohydrates. We also have carbohydrates that are involved in some structures, especially in plants. If we want to store energy, we use the lipid molecule. And the other important function of lipid molecules is to form membranes, boundaries in cells. Any job that needs to be done, any structure that is there, we're going to use a protein, especially enzymes that speed up chemical reactions. And the information to build those proteins is stored in the nucleic acid DNA, and it's turned into a protein using the other nucleic acid, RNA. Then we jump to the cell, and the cell itself was what we know about the cell was very closely related to information we'd gained through the use of a microscope to understand the cell theory. We talked about the cell theory, and then we did talk briefly about the difference between a prokaryotic cell and a eukaryotic cell. Remember, prokaryotic cells do not have membrane-bound organelles, while eukaryotic cells do. We have spent most of the year discussing eukaryotic cells. So that cell theory states that all living organisms are composed of 1 or more cells. Cells are the basic unit of structure and function in all living organisms. Cells come from previously existing cells. They don't just spontaneously appear somewhere. And they pass along copies of their genetic material to their daughter cells. So there is a continuity of life from the first cell to the most complex cell we have now. First part of that cell that we looked at was the outside boundary, that is the cell membrane or the plasma membrane. And we described that plasma membrane as being or demonstrating a fluid mosaic model. So it is a model, it's a representation of the cell membrane. It's composed of different pieces, which is why we call it a mosaic. Those pieces are the phospholipid bilayer, the proteins that are embedded in that phospholipid bilayer, the carbohydrates that are found on the outside and inside of that plasma membrane are the third component. And then embedded within the membrane itself are cholesterol molecules that make that membrane stronger, but they also make it more fluid. So that fluid part tells us that all of these components of this plasma membrane are moving around as though they were floating in a fluid. So selective permeability was the property of that plasma membrane that allowed it to control what entered and left the cell. Very important. It allows us to keep the internal environment of that cell at homeostasis, a topic we touched on in Chapter 1, a topic we focused on in Chapter 1. So that plasma membrane is made up of 2 layers of phospholipid molecules, the phospholipid bilayer. Within that phospholipid bilayer, there are cholesterol molecules that make it stronger and more fluid, transport proteins that help with the selective permeability, and again, the fluid mosaic model describes what we understand about membranes at this point. We then talked about eukaryotic cells, and the 2 basic types of eukaryotic cells were plant and animal cells. Animal cells had centrioles, small vacuoles if they had them at all, whereas plant cells had chloroplasts, large vacuoles, and plant cells have an extra membrane around, or an extra structure, it's not really a membrane, around the outside, it's made of carbohydrates, called the cell wall, that we don't see in animal cells. So we can think of a cell, whether it's a eukaryotic, a prokaryotic, whether it's plant or animal, as a factory, a protein building factory. The nucleus, one of the organelles, contain the instructions, and it helps to build the ribosomes. Instructions are held in the DNA. Those ribosomes, which are mostly RNA, turn the instructions into proteins. Those proteins need to be modified, and that begins to happen in the endoplasmic reticulum. It may do things like add complex carbohydrates to them, part of the identity molecules of the cell membrane. From the endoplasmic reticulum, the Golgi apparatus delivers them to wherever they need to be, whether it be the cytoplasm in some cases, the membrane in other cases, the lysosomes, the nucleus. They are sent there from the Golgi in packets. And mitochondria provide the energy to do all of these things to turn out life. So it's really a factory that puts out life, what we described in Chapter 1, and it does so using machines called proteins that are built using instructions written in DNA. If it's a plant cell, it also has the ability to build the fuel required for the mitochondria, and that would be carbohydrates. So eukaryotic cells contain membrane-bound organelles. They are found within the cytoplasm, and they perform specific cell functions. Ribosomes are one of those, which is not membrane-bound, but it is the site of protein synthesis. Mitochondria provide the ATP needed to run the cell, so they are called the powerhouse of the cell. And plant and animal cells have many of the same organelles, the ones we just spoke of, plant and animals both have, but there are some that are unique to either plant or animal. We have chloroplasts in plant, not in animal, centrioles in animal, not in plant. Energy. So how do mitochondria provide the energy for the cell? Well, the first thing we have to remember is that each individual action that happens in a cell happens because of the use of ATP. And ATP can be thought of as a rechargeable battery. When it's charged, we have 3 phosphates attached to it, and we're going to use the energy when this last phosphate is broken off. And when that happens, that phosphate gets attached to something. Maybe it's a transmembrane protein that we want to change the shape of. When the phosphate gets attached to that energy changes what it's attached to. The phosphate comes off ATP, but it stays in the cell, and then it will be put back on the cell, back on the ADP, through the process of cellular respiration that we'll get into in more details shortly. So ATP does the work within the cell minute to minute to keep the cell alive. And it does so by taking that third phosphate, removing it from ATP, leaving you ADP, and energy is used. And then the phosphate can be put back on using energy from carbohydrates. So plants contain chloroplasts. They have light-absorbing pigments. They are capable of converting light energy into chemical energy. Remember, when chlorophyll gets excited by sunlight, it loses a high-energy electron. That high-energy electron is chemical energy, and that high-energy electron is going to be used to build glucose. Photosynthesis is a two-phase process. It consists of the light reaction, in which light is converted directly to chemical energy, and the Calvin cycle, in which carbon dioxides are added together to give you glucose. So in the light reactions, autotrophs trap and convert light energy into chemical energy. The form of chemical energy is either NADPH or ATP. We focused on ATP. In the Calvin cycle, chemical energy and ATP and NAD are then used to synthesize carbohydrates by attaching carbon dioxides to each other and forming glucose. That glucose is used by all organisms for cellular respiration. It may not use it, they all can't use it completely, but they can all do at least the first part of respiration, and that is glycolysis. So all living organisms use cellular respiration, at least parts of it, to break down glucose and make ATP. Now, if we talk about this stages maximum use of glucose to get ATP, then we're going to go through glycolysis, in which which glucose is broken into two pyruvic acid molecules, 6-carbon glucose into two 3-carbon molecules. Those 3-carbon molecules will then go through the Krebs cycle, starting as pyruvate, becoming CoA, acetyl-CoA, and so on. And in the Krebs cycle, we get carbon dioxide released and high-energy electrons. Those high-energy electrons are carried by transporters, NADH and FADH2, to the electron transport system, and the electron transport system makes most of the ATP that we get from glucose. If we don't have enough oxygen, we can use fermentation. I'm not going to go too much into fermentation today. We covered it a lot during the year, but organisms can all do glycolysis. Some organisms stop at glycolysis or convert directly to fermentation then. Others will continue all the way through all 3 steps as long as oxygen is available. So photosynthesis and cellular respiration are metabolic pathways that produce, in the case of photosynthesis, and break down, in the case of respiration, carbohydrates. They are used to capture energy and use energy. They both occur in plants. Plants have the ability to capture sunlight energy and build glucose, and then use that glucose in their mitochondria to make ATP for the cell. In heterotrophs, only cellular respiration occurs. So in heterotrophs, you don't have the ability to take an energy source and convert it to carbohydrates that you can use in mitochondria. So that means that heterotrophs have to eat to get their carbohydrates. And a lot of the last part of the year, we've talked about how that relationship relates to the environments in which they live in. But it's important for you to remember that photosynthesis and cellular respirations are coupled reactions. The products of photosynthesis go into are the reactants of cellular respiration. The reactants of cellular respiration are the products of cellular respiration are the reactants in photosynthesis. So the reactants in photosynthesis produce products. Those products are the reactants used in respiration. The reactants in respiration produce products, and those products are the reactants in photosynthesis. It's really about how energy and matter are moving through a food chain. Autotrophs are autotrophs because they can pull matter, carbon dioxide, out of the air and energy from the sun to build carbohydrates. Heterotrophs have to eat to get the matter that contains the energy trapped from the sun, and those are carbohydrates. The way they eat determines what kind of heterotroph they are going to be, a primary consumer or a secondary consumer, or an apex predator. All right, that covers the first part of the year. Tomorrow I will post a video with the second part of the year. Again, I appreciate all of you that are watching these. I see the analytics on YouTube, so I know some of them are not being watched, some of them are. I'm just thankful for all of you that are out there watching them. It really is appreciated. Let's get this last week out of the way and hope for a more normalized year next year. Thank you.